Imagine for a second that you go to the doctor and they take a standard, like incredibly cheap, everyday photograph of some tissue.
Right, just a basic medical image.
Yeah, exactly. The kind they've been taking for 100 years.
Yeah.
But when you look closely at this image, it magically reveals an invisible underlying genetic blueprint of every single cell.
Oh, wow.
Yeah. It shows exactly which genes are turned on, which are turned off, and what they are like actively doing in real time.
I mean, that sounds completely like science fiction.
It really does.
Because in the real world, seeing that level of genetic detail requires incredibly expensive, specialized laboratory equipment. And usually a lot of waiting. You certainly don't just get it from a standard picture.
Right. But that is exactly what we are exploring today. We are looking at this fascinating new research paper that introduces an AI framework called SpaHGC.
Yeah.
And the mission of this deep dive is to unpack how this specific AI is basically cracking the code of spatial gene expression.
Which is huge.
It is. By doing this, it's potentially making some of the most expensive and complex medical tests obsolete.
What's really fascinating here is that the researchers are bridging a massive gap in modern medicine. They are taking something incredibly ubiquitous and, well, extracting a level of dense biological data that is usually reserved for top-tier research facilities.
Okay, let's unpack this. The paper itself is highly technical. It's titled Embrace Yourself: Cross-Slice Knowledge Transfer via Masked Multimodal Heterogeneous Graph Contrastive Learning for Spatial Gene Expression Inference.
Yeah, that is a massive mouthful.
It is, but we're gonna keep it accessible and focus on exactly how this machine works. But before we get into the AI itself, can you kind of give us the lay of the land? Like, why is predicting gene expression such a big deal in the first place?
Sure. So to really appreciate the breakthrough, you have to understand the two very different worlds of medical testing we're dealing with here.
Okay.
On one hand, you have something called spatial transcriptomics, or ST for short.
S2, got it.
Yeah. And ST is a breathtaking technology.
Yeah.
It doesn't just, like, put a tumor in a blender and tell you what genes are active overall.
Right, it doesn't just give you an average.
Exactly. It maps exactly where they are active. It gives you this high-definition geographic map of gene expression across a slice of tissue.
Which is absolutely huge for understanding things like tumors, right? Because a tumor isn't just one uniform homogeneous blob.
Not at all.
Like, the cells on the outer aggressive edge of a tumor are doing vastly different things than the dying cells in the center.
You hit the nail on the head. That spatial context is everything for targeted treatments. But the massive hurdle is that ST protocols are incredibly expensive.
Like, how expensive?
We're talking sometimes thousands of dollars per slide.
Wow.
Yeah. And technically, they are notoriously difficult to run. Because of that cost and complexity, you simply cannot use ST on every single patient in a large-scale clinical setting. It creates this major diagnostic bottleneck.
Which brings us to the second world, the cheap stuff.
Yeah, the cheap stuff, right. The second world is the standard H&E stained pathology image.
H&E?
Yeah, H&E stands for 𝗧𝗎𝑚etoxylin and eosin.
Okay.
It's a tissue staining technique that has literally been around for over a century. It uses pink and blue dyes to highlight the physical structures of the cells. So, like the nuclei, the cytoplasm. So pathologists can actually look at them under a microscope and spot abnormalities.
And these are pretty common, right?
Oh, they are everywhere. These H&E images are acquired in routine clinical workflows every single day in almost every hospital on Earth. They cost pennies, they are fast, and they're everywhere.
So the holy grail here is essentially a translation problem. Can we predict the highly expensive, rare ST data just by looking at the incredibly cheap, ubiquitous H&E images?
That is the ultimate goal.
Yeah.
Because each spot on an expensive ST map perfectly aligns with a physical patch of tissue on the cheap H&E image.
Right, they're looking at the same real estate.
Exactly. There's a natural structural foundation linking them. So the theory is, if an AI can learn the hidden relationship between the visual shape and structure of the cells in the cheap image and the complex gene expression in the expensive test, it completely changes the entire landscape of diagnostics.
But wait, I have to push back a little here.
Go for it.
If these standard pink and blue pathology images are so common, and we have AI models right now that can literally, drive cars through chaotic city traffic or instantly generate photorealistic art.
Sure.
Why can't existing AI just look at millions of these medical slides and brute force the pattern? Like, why hasn't this been solved by just throwing computing power at it?
It's a very logical assumption. And to understand why brute forcing it completely fails, we need to look at how previous AI models have tried to tackle this and basically hit a wall.
Okay.
There have basically been three main computational approaches before SpaHGC came along.
So if we have these cheap images and we know the genetic map exists, how did early AI try to bridge the gap? I imagine they just looked at a tiny piece of the image and tried to guess the genes.
You are describing the exact premise of the first major approach, which we call image-to-expression mapping.
Oh, okay.
A prime example is a model called STNet. These models are pretty straightforward. They chop the H&E image into tiny local patches, run each patch through a neural network, and just try to predict the genes for that specific isolated spot.
Just staring at the pixels right in front of them, completely ignoring what's happening next door.
And that is their downfall. Because in biology, no cell acts alone. A cell's behavior is heavily influenced by its microenvironment.
Like the immune cells or blood vessels right next to it.
Exactly. By only looking at isolated local patches, these models are essentially trying to understand the plot of a movie by looking at a single frame. They completely miss the broader spatial context of the tissue.
Okay, so approach number one is way too short-sighted. I'm guessing the obvious fix was just to make the AI look wider, like take in the whole neighborhood?
That was the second attempt. It's known as intra-slide spatial modeling, using models like histogene and his2st.
And how did they work?
Well, they introduce graph networks to look at a spot and its surrounding neighbors. They allow a sort of message passing between adjacent areas, so the AI finally understands those local spatial dependencies.
Okay, that sounds like a massive improvement. Now the AI knows it's looking at a neighborhood, not just an isolated house.
It is a significant step forward, but it carries a fatal flaw.
Which is?
It only looks within a single tissue slice. It is completely blind to the outside world. It cannot effectively leverage shared patterns across different tissue samples from different patients.
Ah, I see. And I imagine every single patient's tumor is wildly different.
Vastly different. This is the core bottleneck of individual variability. 10 individuals with the exact same type of breast cancer might share some broad visual similarities, but the heterogeneity,
The individual differences.
right, the unique individual differences in how their disease progresses at a microscopic level is just staggering.
Wow.
So if the AI is only looking within a single slice, it can't learn the generalizable, overarching rules of expression that exist across a broader human population. It literally misses the forest for the trees.
So we have AI that's either ignoring the neighbors or it's trapped in its own little town, oblivious to the rest of the world. What was the third attempt?
The third is multimodal alignment with models like 𝗬𝗏 and EGN.
𝗬𝗏 and EGN, okay.
Yeah. These attempt to force the image features and the gene features to align into a shared mathematical space. But again, while they get better at local structural modeling, they fail to effectively integrate that information across different patients. They lack a true, functional, global context.
Which brings us to the hero of our deep dive, SpaHGC.
Yes.
How does it break out of this trap? Like, how do you teach a computer to see the local structure of one patient's tissue while simultaneously comparing it to the global patterns of hundreds of other patients?
By building a completely new kind of map. The core innovation of SpaHGC is right in its name. It constructs a multimodal heterogeneous graph.
We promised to keep this accessible, so let's translate that. A graph in computer science isn't like a bar chart, right? It's basically a vast network of nodes connected by lines or edges.
That's a perfect way to visualize it. Think of the nodes as the distinct points of physical data on the slide, and the edges as the defined relationships between them.
Okay.
SpaHGC constructs a massive network using 3 very specific, distinct types of connections or edges to overcome every limitation we just discussed.
Okay, let's break down the architecture. Edge type number 1.
The first type is the target slice edge, or TS edge. This connects neighboring spots strictly on the target patient's actual slide.
So this is handling the local neighborhood problem. It guarantees the AI knows what the cells immediately next door are up to.
Exactly. It captures the vital local spatial continuity. But we just established that local isn't enough. So they introduced the second type of connection, the reference slice edge, or RS edge.
And how does that expand the map?
It completely leaves the target patient. It connects reference nodes to other similar reference nodes pulled from a massive database of other patients.
Oh, wow.
Yeah, it builds a distinct global understanding of gene expression within a massive reference cohort. It establishes the baseline biological rules for how this tissue behaves across humanity, not just in one person.
Okay, I'm tracking with you. We have a local network mapping the target patient and a massive global network mapping the historical reference data.
Right.
But right now, they're totally separate islands. How do they actually talk to each other?
That is the third connection, and it is the vital bridge of the entire framework. They are called cross-slice edges, or CS edges.
Okay.
This explicitly connects the target patient's specific tissue spots to the top 7 most similar reference spots from other slides in the database.
Wait, hold on. How does the system know which 7 spots from a database of thousands of other patients are the most similar if it doesn't even know the gene expression of our target patient yet?
That is where the multimodal part of the title comes in. It uses the visual image data as the initial translator.
Okay, how so?
The researchers utilized a specialized, pre-trained pathology foundation model called UNI.
Wait, how is UNI different from, like, the AI that recognizes faces on my phone?
Well, general vision models, like ResNet 18 or VIT, are trained on millions of random internet images.
Yeah.
Dogs, cars, coffee cups.
Right.
UNI is a foundation model trained specifically on massive amounts of highly complex medical pathology slides. When tested, UNI significantly outperformed those general models because it actually understands the language of cellular structures.
Oh, that makes sense.
Yeah, so UNI extracts deep, nuanced visual embeddings from the target patient's cheap H&E image. The system then queries the global reference database to find the 7 spots from other patients that look visually the most identical to the target spot.
So imagine you're at a massive, overwhelming medical conference.
Okay.
The target slice edge is you talking to the doctor sitting right next to you at your specific table, getting a feel for the local room. The reference slice edge is the world's top experts mingling and sharing their vast historical data with each other behind closed doors in the VIP room.
I like this.
And the cross slice edge is you being able to pull out your phone, snap a picture of a weird case, and instantly text the top 7 experts in the VIP room who have seen cases that look exactly like yours, asking for their genetic data. You're combining your local context with their global expertise.
If we connect this to the bigger picture, this specific graph structure allows for what they call inter-slice knowledge transfer. The vast historical knowledge from the VIP room flows directly to your specific table.
That's incredible.
By linking the target patient to multiple established references based on visual similarity, the AI suppresses the chaotic noise of individual patient variability and hones in on shared underlying biological truths.
Okay, I love the conference analogy, but here's where it gets really interesting.
Oh, yeah.
Drawing lines between data points is one thing, but how does the actual information flow across these bridges without turning into a chaotic mess?
That's the real challenge.
Because biological data, especially at this microscopic level, is incredibly noisy. If the experts in the VIP room are all shouting advice at once, how does the target patient make sense of it?
The researchers anticipated that exact chaos. And this brings us to the internal machinery of SpaHGC, specifically 2 modules that act as the intelligent traffic controllers for this information flow, CNDA and CNAP.
Okay, let's start with CNDA. How does it filter the noise?
CNDA stands for cross-node dual attention. It operates specifically on those bridge connections, so the text messages to the VIP room.
Right.
It's a bidirectional communication channel powered by an attention mechanism. And in AI, attention basically means the model learns what parts of the data to focus on and what to ignore.
Bidirectional meaning the target patient and the VIP reference expert are actively examining each other.
Precisely. The target node looks at both the visual and genetic data of the reference node, and simultaneously the reference node evaluates the visual data of the target node. Through this dynamic dual attention mechanism, they selectively transfer only the most crucial knowledge. The target node essentially says, based on the nuanced details of what my cells look like, which specific pieces of your historical genetic data are actually relevant to me.
That's brilliant. It's not just a blind data dump. It's a selective, highly intelligent conversation.
Exactly.
And what about the second module, CNAP?
CNAP is the cross-node attention pool. Once the CNDA has successfully gathered this highly curated auxiliary information from the 7 reference experts, CNAP dynamically pools it all together. But here's the critical design choice. It is explicitly engineered to ensure the target node relies heavily on its own unique visual representation.
Wait, why is that a critical choice? If these reference nodes are the ultimate experts, why not just copy their genetic answers completely?
Because if it just copied the experts, it would completely overwrite the unique individual characteristics of the patient sitting in front of us.
Oh, right.
Remember, every single tumor is uniquely heterogeneous. CNAP forces the model to integrate the reference information as a highly educated guide, but ultimately, the AI must base its final prediction on the actual physical context of the target patient. It prevents the model from just blindly memorizing and copying reference data.
Okay, I follow the logic there. But I have a major question, a serious bit of pushback here regarding a training method the researchers used.
Okay, let's hear it.
They discuss a complementary masking strategy as part of their contrastive learning approach.
Ah, yes. The contrastive learning aspect is where the model really earns its robustness.
Right, but the paper says they intentionally hid or masked 80% of the target node data and a whopping 90% of the reference node data when they were training the network.
Yes, they did.
Now, we just established that spatial transcriptomics data is already notoriously sparse, full of missing values, and incredibly messy. If the data is already that bad, isn't hiding 90% of the usable information completely counterproductive? Like, why intentionally blindfold the AI?
I love this question because it touches on one of the most powerful and sort of counterintuitive concepts in modern machine learning. By intentionally destroying and hiding that data during training, they're actually forcing the model to become vastly more intelligent and resilient.
Walk me through that, because it sounds like making a student study for a test by burning their textbook.
It's more like teaching a student to understand the core principles of mathematics, rather than just memorizing the answers to a specific worksheet. They generate 2 drastically different augmented views of the exact same tissue data with different massive chunks missing, and force the AI to mathematically realize they're actually looking at the same underlying object.
Okay.
This masking specifically simulates the severe dropout and sequencing noise you encounter in real, messy biological experiments.
So by forcing the AI to reconstruct the underlying patterns and make accurate predictions with only like 10 to 20% of the data visible,
the AI can no longer rely on memorizing surface-level noise or simple shortcuts. It is mathematically forced to learn the deep, fundamental structural rules of the biology. It's like a professional runner doing all of their training with heavy lead weights strapped to their ankles.
Wow, okay.
It makes the daily training agonizingly difficult.
But when you take the weights off for the actual race...
They absolutely fly. When you remove that 90% masking during actual real-world use in a clinic, the model is incredibly robust. It has learned what truly matters structurally, so the inherent messiness of a real hospital's data doesn't phase it at all.
That is a wild concept, but the ankle weights analogy makes it click perfectly.
Yeah.
So let's talk about the actual race. We know how the machine works. We know the brutal way it trained. How well did it actually perform when they let it loose in the lab?
The results are frankly quite staggering for this field. They didn't just test it on one easy sample. They benchmarked SpaHGC on 7 different highly complex public data sets.
Like what kind of tissues?
We are talking about diverse, difficult tissues. Human skin, squamous cell carcinoma, which they abbreviate as CSCC, along with lymph nodes, pancreatic cancer, and highly variable breast cancers.
And breast cancer alone is a massive challenge, right? The paper mentioned HER2+ and TNBC.
Exactly. TNBC stands for triple-negative breast cancer. It's notorious because the cancer cells don't have the 3 common receptors that we normally target with treatments, making it incredibly aggressive and hard to fight.
Right.
Accurately mapping the spatial genetics of a TNBC tumor is vital for finding new ways to attack it. Testing the AI on such wildly different tissue architectures is a real gauntlet.
And they compared SpaHGC against those 9 earlier baseline models we discussed.
Yes, and SpaHGC significantly outperformed every single one of them across all evaluation metrics.
Let's get into the numbers. What kind of improvement are we actually seeing?
So one of the key metrics they use is the Pearson correlation coefficient, or PCC. Without getting bogged down in the math, it basically measures how tightly the AI's predicted spatial map matches the actual wildly expensive ground truth map. Okay. SpaHGC improved the PCC by a massive 7.3% to 27.1% over the absolute best existing models. That is a generational leap in accuracy.
And what about speed? Because if this requires a supercomputer running for 3 weeks to process one patient's image, it's never going to be adopted in a busy hospital.
This is perhaps one of the most practical victories of the paper. Despite having 114 million trainable parameters making it a deeply complex model, it was actually the fastest model tested on a standard NVIDIA RTX 4090 GPU.
Wait, which is just a high-end consumer graphics card, right? The kind PC gamers use, not a multi-million-dollar server rack.
Exactly. Running on that commercially available card, it processed the entire skin cancer dataset in just 1.68 hours.
Less than 2 hours on consumer hardware.
Yeah.
So what does this all mean for you?
For you, the listener, this points to a near future where a routine, incredibly cheap biopsy could immediately give your doctor a highly personalized, high-resolution map of exactly how aggressive your specific tissue is. No waiting weeks for specialized labs, no astronomical bills for genetic sequencing. It democratizes access to top-tier personalized medicine.
That is the real-world impact right there. But does the AI actually understand the biology, or is it just really good at matching pixel patterns?
It genuinely translates to the biology. SpaHGC proved capable of perfectly reconstructing highly complex, specific gene expression patterns.
Oh, really?
Yeah. For example, it accurately mapped the spatial layout of the CNK5 gene in skin cancer and the C8CAIM5 gene in pancreatic cancer. When you look at the visual heat maps published in the paper, SpaHGC's predictions look nearly identical to the ground truth, whereas the older models just produce a blurry, unreadable mess.
And it was even able to find the hidden boundaries of the tumors, right?
Yes, this is a huge clinical application. They use the predicted gene expressions from SpaHGC to cluster the tissue spots into functional groups.
Okay, when compared to the gold standard annotations meticulously drawn by actual human pathologists, SpaHGC achieved the highest adjusted RAND index, or ARI score. It was definitively the most accurate at distinctly separating the dangerous tumor regions from the healthy tissue, purely based on the invisible genetic data it inferred from a basic photograph.
That feels like the ultimate validation of the technology. But the researchers went one step further. They took these predicted expressions and ran them through biological pathway enrichment analyses, specifically looking at massive digital biological encyclopedias like KG and 𝗏
Right. They wanted to see if the individual genes the AI predicted actually function together in known, coordinated biological systems. It's one thing to predict a letter, it's another to write a coherent sentence.
And the AI wrote the sentence. The predictions mapped perfectly to known biological realities, like the B-cell receptor signaling pathway, which is heavily involved in immune response and tumor progression.
The AI isn't just playing a matching game to get a good math score. It is successfully inferring actual functional biological pathways just from the subtle shapes and structures of the cells in a standard pink and blue photograph.
This raises an important question.
It really does. Because if we recap this incredible journey, we started with a massive problem. Getting a high-resolution map of exactly what genes are doing inside a complex tumor is vital. but currently it is way too expensive and complex for routine clinical use.
And we saw how previous AI attempts hit a wall because they either ignored the local neighborhood context entirely or they got trapped inside a single tissue slice, unable to learn the shared biological rules across thousands of different patients.
Then we looked at SpaHGC, which cracked the code by building a multimodal heterogeneous graph. It used target slice edges to understand the local cellular neighborhood, reference slice edges to understand the global biological rules, and cross-slice edges to bridge the gap, letting the target patient literally text the top 7 VIP experts for advice using the deep visual understanding of the UNI Foundation model.
We also unpacked how it used bidirectional attention modules, CNDA and CNAP, to intelligently pool that expert knowledge without overriding the patient's unique biological identity.
Right.
And most surprisingly, how it used that heavy ankle weight masking strategy, blinding itself to 90% of the data during training, to build an incredibly robust, noise-resistant understanding of tissue architecture.
And the real-world results speak for themselves. Beating 9 state-of-the-art models, improving accuracy by up to 27%, running in under 2 hours on a gaming graphics card, and successfully identifying complex biological pathways and hidden tumor boundaries. It is a profound leap forward that could change how we diagnose disease.
Absolutely.
And I want to leave you with this final thought. Think about this. If a 100-year-old, incredibly cheap tissue staining technique like 𝗏𝗫 secretly contains the hidden map to our entire complex genetic universe, and AI is just now learning how to read it, what other invisible biological truths are hiding in plain sight in standard medical data, just waiting for the right algorithm to decode them?
It makes you look at a simple medical photograph very, very differently.
It really does. Thank you so much for joining us on this deep dive. We'll see you next time.